When I was in high school, we were taught kingdom, phylum, class, order, family, genus, species. Kingdom, phylum, class, order, family, genus, species. And within the last 20 years, there has been domains added to the top. And there are three major domains of life: bacteria, which we also call prokaryotes or prokaryota, archaea, and eukaryota, which we also call eukaryotes. Okay, so one of the lectures that you should have watched was entirely on the differences between prokaryotes versus eukaryotes. Okay, and if you look at the phylogenetic tree, what goes right here is LUCA. LUCA stands for last universal common ancestor. Okay, so when we say common ancestor, it means if you have an organism X and an organism Y and they evolved, this was their ancestor. So X and Y share a common ancestor. So it's thought that life emerged one time. It's thought that life evolved one time, and that first organism is what we call LUCA, the last universal common ancestor. And then organisms began to evolve and split into the three domains of life. And because we know the differences between prokaryotes versus eukaryotes, today I want to spend time talking about this branch. Okay, branch. In English, you have a tree. Okay, that is a branch. Okay, so in a phylogenetic tree, anytime you have a split, that's called a branch. And I want to talk about the branch point where eukaryotes split from archaea. So eukaryotes include animals, vertebrates, yeast, fungi, and us, humans, we are all these organisms, we are all eukaryotes, okay? And eukaryotes are most closely related to archaea, okay? So if you look at the tree, here's archaea, here's eukaryotes. We are closely related to archaea, and very distantly far off is bacteria, okay? So we are close to archaea. And the hydrogen hypothesis, okay, the hydrogen hypothesis, it asks the question, or it gives an answer. It tries to answer the question, how did the first eukaryote evolve? Okay, so let's review. Let us review characteristics of eukaryotes. Characteristics of eukaryotes. Okay, write it in the chat. What are characteristics of eukaryotes? Write them in the chat. What are characteristics of eukaryotes? So this branch, what do they all have in common? What makes a eukaryote a eukaryote? Yes, Yan Lu, thank you. Thank you, Yan Lu. So eukaryotes, let's make a list because one, nucleus. Okay, so this is characteristics of eukaryotes. They have a nucleus, okay? Prokaryotes, no nucleus. What else? What else do you find in eukaryotes? Yes, mitochondria. And in prokaryotes, no mitochondria. Cytoskeleton. Okay, so cells have skeletons made of proteins like tubulin, myosin, actin. These are all cytoskeleton components, and they give structure to the cell. Prokaryotes, no cytoskeleton. Okay. Eukaryotes have long chromosomes. Prokaryotes have short chromosomes. And oftentimes they are circles, whereas eukaryotes often have linear chromosomes like this. So bacterial chromosomes look like this, and eukaryotic chromosomes look like this. What is a big difference about packaging of DNA? Eukaryotes have histones, which package. So package is like when you buy a present for your friend, you put it in a box and you can mail this. A box is what in English we would call it's packaging. It's an analogy. Eukaryotes take their DNA and they package it by wrapping it in histones. Okay, so they wrap it in a protein octamer called histones. Okay, and a histone is an octamer of 8 proteins, and DNA wraps around the histone octamer 3.2 times. So it actually goes 1, 2, 3. Okay, and this structure is called the beads on a string. This is what we call this in English. It's like a pearl necklace that somebody wears, beads on a string. That's what the histone octamer looks like. And this histone octamer, which wraps the DNA, it itself is curled and wrapped into a structure that we call the 30 nanometer fiber. And the reason it's called the 30 nanometer fiber is because the width of this is 30 nanometers. Okay, so this is the smallest, and then this is bigger. And then 30 nanometer fibers are wrapped into chromosomes. And what holds those chromosomes together are proteins that look like this, called SMC proteins. And they look like a clamp, and it's like a claw. It holds it together. Okay, so SMC proteins wrap around the chromosomes. So in eukaryotes package their DNA. In prokaryotes, the DNA is open, loose. So in prokaryotes, the DNA is loose. And in eukaryotes, the DNA is packaged. Other differences. So this is euks versus proks. 6, size. Euks are large cells. Prokaryotes are small cells. Okay. So if you do a size comparison, eukaryotic cells look like this. Prokaryotic cells look like this. Proks have small cells. Eukaryotes have large cells. Let's see if anybody caught this from the lecture. What allows eukaryotic cells to be large? Why can eukaryotes have such large cells? Why can eukaryotes have such large cells? Do you know? Write it in the chat. We're close. Organelles is close. What is the problem of large cells? Okay. The problem of large cells is the surface divided by surface area divided by volume ratio. This is a problem. Let me talk about that. So in a small cell, so let's say a proc cell, a small cell, okay? Nutrients, so let's call this food. Food can pass through the membrane, through porins or channels. And once it gets through, it can be absorbed and it can diffuse throughout the cell to feed the whole cell. Okay. So all parts of the cell can receive food, or it could be chemicals, or it could be ions, can receive food equally through diffusion. Okay. In a big cell, in a really big cell, like a eukaryote, food can diffuse across the membrane through ions and pores and channels. But it's very hard to get food inside of the middle of the cell because diffusion looks like this. Diffusion causes flow of high concentration. That's the symbol for concentration. Okay, so diffusion is the process of moving from high concentration to low concentration. Okay, and diffusion works for small things, but not for large things. Okay, so this part of the cell has trouble getting food. Okay, and you might ask the question, you might someday ask the question, why can't cells be infinitely large? Like, why aren't there cells the size of a building? Why are there no cells the size of a house or a building? Why aren't there cells that are that big? This is the reason, okay? Diffusion, this is the reason, the surface to volume ratio, okay? As the cells, let's write out that equation, surface area. The area of a circle is an equation that is squared, okay? Divided by a volume. A volume is cubed to the third power. Surface area is to the second power. So as cells get larger, as cells grow, the volume gets bigger to the third power, but the surface area only gets bigger to the second power, and 2 is less than 3. So as cells get bigger and bigger and bigger and bigger, this ratio gets smaller and smaller and smaller and smaller mathematically, okay? That is this problem. That is this problem. So if you wonder why do eukaryotes have mitochondria, it's because it solves the diffusion problem. If you load the cell with mitochondria, and mitochondria we know produce ATP, which is like food, okay? ATP can now get all through the cell. Is that clear? Do you understand? Yes or no? Write it in the chat. Do you understand this point? So let's talk more about the hydrogen hypothesis. An interesting thing about this tree, let me pull this tree, bring it down. If you look at eukaryotes, okay, eukaryotes, all eukaryotes have mitochondria. There are some rare cases, okay? There are some exceptions, or what we could call rare cases of eukaryotes that lost mitochondria. But in general, the rule is all eukaryotes have mitochondria, okay? And it's because of this, because eukaryotic cells are very large, okay? So some people think about this, and one hypothesis is that in order to become a eukaryotic cell, you must first acquire a mitochondria. Okay, so if we are thinking about this branch, how does archaea become a eukaryote? In order to become a eukaryotic cell, the archaea must first acquire a mitochondria to solve the surface volume ratio problem. Okay, that's one hypothesis. And so the hydrogen hypothesis tries to explain how that happened, how an archaea acquired a mitochondria. Okay, that is what the hydrogen hypothesis is thinking about. Okay, so let's lay out the points of the hydrogen hypothesis. Okay, the hydrogen hypothesis. In general, you could say the thought is that a methanogen absorbed an alpha proteobacteria, which evolved into a eukaryote. That's the hydrogen hypothesis. This is what it is specifically. A methanogen absorbed an alpha proteobacteria, which evolved into a eukaryote. Okay, so let's let's talk about this. Methanogen. What are methanogens? Okay. Methanogens are archaea. Okay. So methanogens are archaea. And they are special. So let's draw this as a methanogen, which is an archaea. Okay. Methanogens use hydrogen. So they need hydrogen plus carbon dioxide to make energy, ATP, and the byproduct is methane. So CH4, methane. This is how they get their name. Methanogens make methane. Methanogens make methane from hydrogen and CO2. Okay. This is point 1 of the hydrogen hypothesis. Point 2, hydrogen, gaseous hydrogen, it only exists in anaerobic environment. So gaseous, how do I spell it? We'll just say it. Hydrogen gas only can exist in anaerobic environments. Okay, so anaerobic means no oxygen. That's what anaerobic means, no oxygen. Okay, so methanogens usually live. So where do methanogens live? Methanogens live in anaerobic environments where there's no oxygen. Okay, so these are 2 important points in the hydrogen hypothesis. Okay, 3, you need to understand what hydrogenosomes are. Hydrogenosomes. Okay, hydrogenosomes are like mitochondria, similar to mitochondria. They share a common ancestor with mitochondria. Okay, so if you were to draw a phylogenetic tree, let me get a good color. If you were to draw a phylogenetic tree, common ancestor, mitochondria, hydrogenosomes. So hydrogenosomes are kind of like, they're like mitochondria. They're very similar. Okay, and they can produce ATP, so they can make ATP, and they also make hydrogen gas and CO2. Okay, so people think that the common ancestor of the mitochondria and the hydrogenosomes, it probably looked like both. It probably looked like both a hydrogenosome and a mitochondria. So that whatever the common ancestor was of the mitochondria, it could probably make ATP, it could make hydrogen gas, it could make CO2, okay? And what mitochondria do is they can perform respiration, okay? So it could probably also respire. So it's thought that the common ancestor of the mitochondria and hydrogenomes could do all these things, okay? Well, there is a bacteria that can do all these things, okay? Rickettsia is a genus of bacteria, and it's within the alpha proteobacteria. Okay, so I have to introduce some nuance here. What I originally said in the lecture is that Rickettsia can do all these things, make ATP, make hydrogen. That's not technically correct. Rickettsia in some cases is stealing ATP, and I'm not sure that it makes hydrogen. But the idea is that sort of a pseudo-ancestral Rickettsia had a mixed bag of metabolism that could do these things, or at least has some enzymes that allow it to do those things. Okay. So now do you see what the hypothesis is? Let me draw it out. The hypothesis is that a methanogen, which needs hydrogen and CO2, can form a symbiosis with an alpha proteobacteria like Rickettsia that can make hydrogen, it can make CO2, which are what the methanogen needs, okay? And so the thought is that methanogens eventually started to live very, very, very close to alpha proteobacteria because they're making the food. They're making the hydrogen and the CO2 that the methanogen needs to make methane. And after some time, after some evolution, the methanogen absorbs the alpha proteobacteria. So now at this point, the methanogen absorbs the alpha proteobacteria. And it's thought that this is the first eukaryote. Okay, it's a merger between two cells. So just to be clear, methanogens are archaea and the alpha proteobacteria are the bacteria. So to make a eukaryote, it's thought that the methanogen plus the alpha proteobacteria makes a eukaryote. That's the hypothesis. And it's called the hydrogen hypothesis, the hydrogen hypothesis, because methanogens need hydrogen and the alpha proteobacteria can make it. That's why it's called the hydrogen hypothesis. So the first eukaryote might have evolved by a methanogen absorbing an alpha proteobacteria. Okay, let's talk about evidence for this. What is the evidence? Evidence for the hydrogen hypothesis. What is the evidence? Look at this picture. This picture. Okay. These, the dark blobs are methanogens, and these are hydrogenosomes. And remember, hydrogenosomes make hydrogen and CO2, and methanogens eat. They eat it, hydrogen and CO2. So you can actually see, we can actually see this process. There's pictures of it right there in cells. So in cells, in big eukaryotic cells, you can find hydrogenosomes right next to methanogens in the cell. Okay. Now, so that's one piece of evidence. Okay. More evidence. Evidence. Point 2, molecular phylogenetics. So mitochondria have their own genomes. Let me write this out. Mitochondria have a chromosome. They have their own DNA. Their own DNA. Okay, so if this is DNA, mitochondria have their own DNA. And if you compare, if you ask the question, what is the closest organism that matches mitochondrial DNA? So if you look at the mitochondria genome, you look at all the bases, A, T, G, C, on and on and on and on and on to N, and you compare that to all other organisms, it matches alpha proteobacteria. Specifically, it matches very closely to Rickettsia. Okay, so if you do this comparison, they match. They match. They are similar. So that suggests, this is evidence that mitochondria evolved from alpha proteobacteria. This is the evidence, okay? Now there's more evidence. If you take the eukaryotic DNA, okay, so eukaryotic, let's give it a different color. Let's say it's blue, okay. If you take eukaryotic DNA and you do the same thing, A, T, G, C, on and on and on and on, the genome of eukaryotes, and you say, what is the closest organism in archaea that matches eukaryotic DNA? What do you think it is? And people do these comparisons. What do you think it is? What organism? Write it in the chat. Yes, very good. Yahoo. Methanogens. So if you do this comparison, very good. If you do this comparison, the answer is it matches methanogens. This is why we think that methanogen absorbed an alpha proteobacteria to become the first eukaryote. Okay, very good. Now, this is a bit hard to understand, a bit complicated, but I want to talk about it. How is the hydrogen hypothesis different from other hypotheses for eukaryote evolution? How is the hydrogen hypothesis different? How is it different from others? Some of the other, all the hypotheses involve absorption of an archaea and a bacteria. Okay, so I'll type it. All hypotheses involve an archaea absorbing a prokaryote. But the hydrogen hypothesis is the only one that explains this process with symbiosis. Okay, the key word here is symbiosis. The hydrogen hypothesis is the only one that explains this with symbiosis. Okay, the idea that what the methanogen needs is being made by the alpha proteobacteria. There are other hypotheses. The other hypothesis is that maybe what was happening was that the prokaryote was making ATP, and the methanogen, maybe the methanogen wanted ATP. This is an other hypothesis, but scientists don't like this. Scientists don't like this hypothesis. Okay, for three reasons. One, methanogens can already make ATP. Okay, methanogens make their own ATP perfectly fine. Okay, so they don't need ATP. They can already make it. They can already make ATP. So they don't need an alpha proteobacteria to make them ATP. Whereas they cannot make hydrogen. In contrast, or in comparison, they cannot make hydrogen. Okay, so in the hydrogen hypothesis, the symbiosis, they're getting something that they cannot make. And this other hypothesis that says, well, maybe ATP was what they were getting. It doesn't make sense because one, methanogens can already make it. Two, it assumes that the prokaryote had extra ATP. Okay, this hypothesis assumes that the prokaryote had extra ATPs, like it had extra food. Nowhere in nature is there extra food. Okay, it's very hard to survive. No organism except maybe humans has extra food. Okay, so this is highly unlikely. Three, it assumes the alpha proteobacteria would secrete the ATP. Okay, so this assumes in most bacteria, they make ATP inside the cell. Okay, this hypothesis assumes the alpha proteobacteria would spit it out. It's like throwing money away. It's like taking money and like lighting it on fire. It's like burning money. Nobody does that. You don't do that. Okay. So scientists don't like this hypothesis for these three reasons. And the hydrogen hypothesis is much better because in this case, it's the waste products that are useful. It's the waste that is useful for the methanogen. So nobody's losing any energy, nobody's losing any food, nobody is losing any resources. It's the waste that is useful. That's the symbiosis, okay? So that's why we like the hydrogen hypothesis. Okay. Final thoughts. If this is true, we don't know. It's our best hypothesis. If this is true, where did this happen on Earth? Where? We know it has to be in an anaerobic environment. Okay. Take a guess. Take a guess. Where do you think this happens? Yes, very good. Excellent. In the ocean, in the sea, or the ocean. Okay. So if you go out in the ocean and you look way, way, way down in the depths, right? There are, in the Earth's core, there is hot lava, or also called magma, okay? And magma will spill out and it will produce heat. And we call these hot vents. And in these environments, there is no or not much gaseous oxygen, okay? So it is anaerobic. Check. And if you look, okay, so if you look at the organisms that live in this area, you can find methanogens. Methanogens live in these hot vents. So the best guess is that we think this happened in the sea by hot vents. Okay. Final thought. How could we prove this? How could we test this hypothesis? Any thoughts? How could we prove this? How could we test this hypothesis that it happened specifically by these hot vents? How could we test this? Well, Jupiter, so the planet Jupiter has 3 ice moons, okay? The names of these moons are Europa, Ganymede, and Callisto, okay? So there's 3 ice moons on Jupiter called Europa, Ganymede, and Callisto, and they are made of water, okay? And as they go around Jupiter, as these moons orbit around Jupiter, the orbit goes around the planet in a way that when the moon is close, Jupiter's gravity actually stretches the moon. Okay, so if you look at the moon, it stretches. So when it's out here, it's like a perfect circle. But when it's here, Jupiter's gravity stretches the moon. And it's thought that this might heat up the core. It might heat the core. And so if we might be able to test this hypothesis by exploring the oceans of Jupiter's ice moons. So NASA, the National Aeronautics and Space Administration, is planning to send robots to Jupiter's ice moons to look for hot vents and look for life. And maybe we will find eukaryotic life. Maybe. That's one test. This is a hard thing to test, but we're going to try. Maybe it'll be in the next 50 or 100 years, but that's a test. That's the hydrogen hypothesis.
I shall meet death head on. Decker Cage, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The time has come to cleanse my horde. I shall